left off at phosphoglycerides, or a general term for those are phospholipids. They're lipids that have a phosphate group on them. Okay, so it's any lipid that contains phosphorus is considered a phospholipid or a phosphoglyceraldehyde or phosphoglyceride. Phosphoglycerides contain, okay, glycerol. Remember, glycerol is our 3-carbon structure where the OH, there's 3 OH groups, and each one gets replaced with a ester with a long fatty acid chain. Okay, so glycerol, fatty acids, and a phosphoric acid with an amino alcohol. Those are the components of a phospholipid. Okay. Replace an N fatty acid of triglyceride with a phosphoric acid that's linked to an amino alcohol. Okay. So this is glycerol, the same thing we've all been doing, 3 carbons. We still have 2 fatty acid chains, and now we take 1 chain, and we put phosphoric acid on it and attach an amino alcohol to it. And that's your phospholipids now, because there's a phosphate group on it. Okay. Phospholipids are a group of lipids that are phosphate esters. That's because of how they link together, because of the condensation between a carboxylic acid and a alcohol. And that forms an ester. The presence of a phosphor group, okay, results in a molecule with a polar head. Okay, so now we got a polar bond in there because the phosphate group has a charge. So you got a polar head, which is the phosphor group, and a nonpolar tail, which is the long chain fatty acids. So you got 2 ends now, polar end and a nonpolar end with a phospholipids. Because the phosphor group ionized in the solution, a charged lipid results. Okay, so you get an ionic charge on that. So you have a hydrophobic end, which is your long chain fatty acids, and a hydrophilic area, which is your phosphate group. Okay, they make up the structural component of membranes. It's a phospholipid bilayer membrane. They're emulsifying agents. Remember, they can get rid of dirt and stuff because one end is hydrophilic, one end is hydrophobic. The hydrophobic end bonds with oils and stains and grease and things like that because those are hydrophobic. And the polar end bonds with water or other polar molecules. So you can emulsify different things because you have 2 different types of regions on the same molecule. Suspended in water, they spontaneously arrange in ordered structures. So if you just put the phospholipids in water, they're going to form basically a membrane, just like the cell membrane. Okay, because you have a hydrophobic groups go into the center. Hydrophilic groups go towards the outside of the water. So there's a basis of a membrane structure. So if this is your positive end, your charge, and these are your hydrophilic, what they're going to do is they're going to arrange kind of like this, where everything these are your charges. This is your phosphate groups on the end. Okay, so this part is going to be compatible with water. Okay, and this is going to be your hydrophobic group on the inside. So it's going to form what's called a micelle, just automatically, and it forms just like a membrane would form. Okay, the phosphoamino alcohol is hydrophilic. It's got a nitrogen group. Okay, so it's an amine and an alcohol group. They're both of those cause hydrogen bonding. Both of those are highly electronegative, and they will be polar. Okay. They're used in cell membranes, which you said, phospholipid bilayers. They're used in emulsifying agents like in soaps, which you talked about. And they form micelles, just like we drew the last time. Those are those circles that form, okay, in any kind of polar liquid, like plasma in the blood. Okay? They form these little micelles that can travel through polar liquids. There's 2 types of phospholipids. Okay, there's ones made with choline, are called lecithin. Those made with ethanolamine or serine are called cephalins. So you got lecithins and cephalins. Okay, this is phosphatidate. You can see this is your glycerol structure here. These are your ester groups with your long chain fatty acids. And this is your phosphate group. This is your polar end. So you got a polar end, hydrophilic, and a nonpolar end, hydrophobic. Okay. This nonpolar end forms the inside in the micelles, and this forms the polar end forms the outside. So therefore, okay, same thing here. This is your phosphatidylcholine, which is your lecithin, and again, this is your glycerol. Your two ester groups of acids for fatty acid chains. Okay. And then this is the amine alcohol group that forms. Same thing again. This is your cephalin. Again, this is your glycerol group. Two ester groups attached, long chain fatty acids, and then your nitrogen group. Okay, and this is phosphatidylserine. Okay, this is a serine group that attaches to your phosphate group right here. Okay, so lecithins possess a polar head and a nonpolar tail. That's your phospholipids. So it's an amphipathic molecule. Okay, amphipathic means it has both hydrophobic and hydrophilic regions in the same molecule. That's what amphipathic means. It's similar in structure to soap and detergent molecules. That's why it forms on micelles in plasma and other polar liquids. Okay. The ionic head is hydrophilic and interacts with the water molecules. The nonpolar tail is hydrophobic, interacts with the nonpolar molecules. So that's just how soap works and everything else. Okay. The amphipathic nature is central to the structure and function of cell membranes. Remember, cell membranes are your lipid bilayer where you have your polar end and then your nonpolar ends. This is how your membranes form. Okay, these are your polar ends here. And your nonpolar ends, that's what forms your cell membrane. Let's see where we at. Okay. So in addition to being a component of cell membranes, lecithin is a major phospholipid. Surfactant is the phospholipid that coats the lungs when an infant is born. We talked about that already in this class? And we talked about it in anatomy. But surfactant is, when an infant's born, it's got to open the lungs up because they haven't been using their lungs. And think of a balloon. Okay, a balloon is collapsed. How hard you have to blow to get that to open up at first? You've got to blow really hard to push that balloon back apart. But once it's up, once the balloon's inflated, then you can deflate a little and inflate, deflate a little and inflate. It doesn't take that burst of energy like it did at first to open it up. That's what surfactant does. Okay, so surfactant coats the lungs with its phospholipid layer, so the lungs don't collapse. So when the infant breathes, it doesn't have to continue to expand its lungs and open up. That's why premature babies often are going to respiratory arrest because they don't produce surfactant yet. Right? It's also found in egg yolks and soybeans. It's used as an emulsifying agent in ice cream to suspend nonpolar molecules in a polar solution. Okay, an emulsifying agent aids in the suspension of triglycerides in water. Remember, triglycerides are fats. Fats are not dissolved in water. But an emulsifying agent, okay, aids in suspending those into water with a phosphate group, so it has a polar end to it. The amphipathic lecithin serves as a bridge, holding together the highly polar water molecules and the non-polar triglycerides. Okay, remember, one end of the lecithin is polar, it'll bind with water. The other end is non-polar, and it'll bind with the triglycerides. Okay, so emulsification occurs because a hydrophobic head of lecithin dissolves in water, and the hydrophilic head of lecithin dissolves in water, and the hydrophobic tail dissolves in the triglycerides. So they will suspend them even though they are different. They're one's polar, one's non-polar. So you can use the lecithin as a emulsification agent. Okay. Cephalin is similar in general structure to lecithin. The amine group bonded to the phosphorus group is the only difference. So it just depends how the nitrogen group bonds to it. Okay. Non-glyceride lipids. Those are sphingolipids. Those are not made from glycerol. So they don't have that 3-carbon background like the other ones do. Okay, they're based on a different molecule. These are your sphingolipids. These lipids are based on sphingosine, not glycerol. Remember, glycerol is just your 3 carbons. Sphingosine is a different configuration. We'll show you that in just a second. Okay, these sphingolipids have long chain fatty acids. They're nitrogen containing, and they're alcohols. Okay, they're amphipathic, just like the phospholipids. They have a polar end and a non-polar end. Okay, the polar head group, they have a polar head group. They have 2 non-polar fatty acid tails, and they're the structure component of your cellular membranes. These sphingolipids. Major categories of sphingolipids are, they have 2 types. There's sphingomyelins, and there's glycosphingolipids. So you got sphingomyelins and glycosphingolipids are the 2 main categories of the sphingolipids. Okay. This is sphingosine. Remember we had the regular glycerol, just 3 carbons. This has 3 carbons also, but now you got a nitrogen here and NH3 instead of just a hydrogen. And you're missing your OH groups from here. You're missing that one OH group. And then this is your long chain fatty acid. So this whole unit, this is sphingosine. This is what the sphingolipids are based upon, rather than glycerol. Okay. Different types of sphingolipids, like we said, there's sphingomyelins. Sphingomyelins are the structural lipid of nerve cell membranes. That's where they're found. Myelin sheath that goes around the axons of your nerve. Okay. And then you have your glycosphingolipids. Let's go back here. The sphingomyelins. Okay, remember these coat the nerve cells. So if you have your nerves, that's what this is, your nerve cell body, and information comes into it here. And then these have these long axons that come out of them. And this impulse, electrical impulse, travels down the axon. Now it has a certain speed that it goes at. Okay. When you have sphingomyelins, myelin coats this axon with like a myelin sheath. And it's an insulation basically to the nerve. So now when the nerve depolarizes, it fires here and then it skips where the myelin is and it fires again and it fires again. So it doesn't have to travel along those areas where the myelin is. So the nerve impulse travels extremely fast, thousands of times faster than if the myelin wasn't there. Because some of our axons don't have myelin. But the ones that have to act extremely quickly, those are myelinated with these sphingomyelins, and allows the nerve impulse to travel extremely fast, thousands of times faster than it would just on a regular nerve. So that's why these sphingomyelins are real important. And you have glycosphingolipids. Okay, they're built on ceramide. Okay, cerebrosides have a single monosaccharidated head group. So they got a sugar group at the top, at the head position of the molecule, and it's a monosaccharide, glucose, galactose. Okay? It's just a 6-ring or 5-ring sugar that have the group. Okay, glucocerebosides are found in the membranes of macrophages. Macrophages, we men, are phagocytic cells. They eat other cells, eat pathogens, viruses, bacteria. Okay, that's where your glucocerebosides are found, in the membranes of macrophages. Galactocerebosides are found almost always in the membrane of your brain cells. So in the glucocerebosides, glucose, okay, is the monosaccharide head. In galactocereboside, galactose is the monosaccharide head on that group. Okay. Remember, glucose and galactose, the only difference, they're both monosaccharides. The only difference between them is that at the number 4 carbon, the OH group is up, or the OH group is a different orientation. That's the only difference between glucose and galactose, is that the OH group in the number 4 position on the ring. So the sphingolipids include the sphingomyelins and the glycosphingolipids. The sphingomyelins we talked about are the only class of sphingolipids that are also phospholipids. Okay? Those are the ones that coat the myelin around the central nervous system. Okay, so here's your sphingomyelin. Okay, this is sphingosine is this right here. This is our group. This is the backbone. Just like glycerol is the backbone of the other ones, sphingosine is the backbone for this one. To attach that, we have our ester, okay, fatty acid. Okay, we have our phosphate group, and we have a choline group. This is sphingomyelin. That's what coats the axon sheaths for as the axons travel across the body to signal that impulse. All right. So the sphingomyelins looking at the body, but are particularly important. Okay, and the components of the nerve cell membranes, just like we talked about. They're found in abundance in the myelin sheath that surrounds and insulates cells of the central nervous system. So within the central nervous system, your brain and your spinal cord, the cells that run through there transferring information, those fibers, those axons are coated with myelin. So they can transfer information extremely quick, much faster than if they weren't. Okay? In humans, about 25% of the lipids in the myelin sheath are these sphingomyelins. Okay? They're essential to proper cerebral functioning and nerve transmission. So they're essential for your brain to function correctly and to transmit the correct signals. All right. Glycosphingolipids or glycolipids include the cerebroside sulfatides and gangliosides. Okay, they're built on a ceramide background. A ceramide background, okay, is a fatty acid amide derivative of sphingosine. So it's just like the sphingosine that we saw, but it's a little bit different. It's a derivative of it. Okay. So here's your backbone again. Okay. That is ceramide. Okay. You have a fatty acid attachment to it of the nitrogen. Which is your fatty acid right there. Okay. And those are those are the glycolipids. Okay. The cerebrosides are characterized by the presence of a single monosaccharide head group, like we talked about. We said the difference between glucose and galactose. Okay. Two common cerebrosides are glucose cerebroside, which found in the membranes of macrophages. Where macrophages are ones that destroy foreign bodies, pathogens, bacteria, viruses. Okay, they go around, they dissolve, invading pathogens. Okay, those are the glucose cerebroside. And then the galacto cerebroside. Okay, those are ones that are found in the membranes of brain cells. Okay, so glucose cerebroside consists of the ceramide bonded to the hexose glucose. Okay, remember the glucose sugar, the hexose, the 6-ring structure of it. And galactose cerebroside consists of ceramide joined to the monosaccharide galactose. Okay. So glucose cerebroside in your macrophages has the glucose monosaccharide on the end of it attached. Okay. Galactose cerebroside has the galactose sugar attached, okay, to the ceramide. Okay. So this is glucose. This is galactose. And remember, we said the only difference was that the number 4 carbon, the position of the OH group. So this is your number 4 carbon. In galactose, the OH group is up. In glucose, the OH group is down. Went right through it that way. Okay, that's your difference. They both have the ceramide structure, just a different sugar on the end. That's the only difference between them. So a glucocerebroside and a galactocerebroside. So just remember, the glucocerebrosides are ones involved in macrophages. Okay. And the galactocerebroside is one found in brain cells. Sphingolipid diseases. Okay, when something goes wrong with the enzymes, okay, these sphingolipids are very important in establishing communication, establishing our brain patterns, expressing pathogens. Now, if we can't break them down and we keep forming them, they build up. Lipids are not a good thing to build up in the body. You don't want to deposit fat all through your body. Okay, that's not a good thing in the brain. Depending on what the disease is, it has different symptoms, like Tay-Sachs disease. Tay-Sachs disease causes blindness, muscle weakness. You usually don't make it past the first few years of life. The sphingolipid is gangliosides, and the enzyme is beta-hexosaminidase A. So it's not being broken down. Gaucher's disease, that's a more common disease. Okay, you'll see that in your clinics when you start working as an RN. Liver and spleens enlarge.
Can you visually tell
Glucose
when a child has
triglycerides.
Tay-Sachs?
Could you tell what?
Can you visually tell when the child has Tay-Sachs? Because I mean, there's some babies that are just like super fat.
By looking at them, no. But you will find that their eyesight will not develop and they will not be able to walk at the normal ages because their muscles won't develop.
Okay.
Tay-Sachs is a European disease mostly, and it's the genetic markers that we tie for it. But we'll go over the differences and some of the things here too on some slides at it. I'll show you one of the MRIs of a brain with a Tay-Sachs versus a brain that does not have Tay-Sachs. Because in Tay-Sachs disease, you store it in the fat deposit is stored in the brain, and it distorts the brain tremendously. You'll see the differences. We'll show some pictures of that in just a second here, because it makes it's much more visual. You see pictures of it, you learn it better. You have Graves' disease, which is demyelination. Okay, demyelination means the myelins doesn't form, the sphingomyelins. Okay, so you don't get the nerve impulses traveling as quick as they should, and the brain does not function correctly. And that's beta galactosidase is the enzyme in that one. And you have Niemann-Pick disease also, which is your sphingomyelin, which is sphingomyelinase, and it's got too much myelin being developed. So those are the basic diseases. Okay. Okay, there are a number of human genetic disorders caused by deficiency in one of the enzymes responsible for the breakdown of the sphingolipids. Like I said, you don't want these sphingolipids develop where you cannot break them down because they got to be stored somewhere. So in general, the symptoms are caused by the accumulation of abnormally large amounts of lipids within particular cells. Each disease has a specific area where the extra lipids are stored. Okay. As you're interested to know, the three of the diseases, Niemann-Pick disease, Gaucher's disease, and Tay-Sachs disease, are found much more frequently among Asiatic Jews of Northern European heritage than among any other ethnic group. Okay, so if your heritage is from an Ashkenazi Jew in Northern Europe, you get genetic testing before you have a baby because it's a recessive gene that carries the Tay-Sachs disease and the other ones. And we'll show you how that works on another diagram too here. How we how it's transmitted generation to generation. Okay, of the four subtypes of Niemann-Pick disease, type A is the most severe. Okay, it's inherited as a recessive disorder. The defective copy of the gene must be inherited from each parent. Okay, recessive, when you have genes, you have a dominant and a recessive. So we have a dominant gene that's a capital R, recessive is a little g. If this is a father, and then a dominant recessive gene, this is the mother. Okay. Yeah, I got that right. Okay. Now, when they mix the genes down there, you got four different types of either one. This father here is a carrier. He does not have the disease. This mother here is a carrier. She does not have the disease. The only way to tell is doing genetic testing. Because when combinations with the chromosomes, the only possible combinations you can have are R, big R, R, little r, or R, again, little r, or little r, little r. Okay, where this big R comes from the parent, from the father, this big R comes from the mother. Okay, so 25% chance of their offspring will have Niemann-Pick disease. That's what recessive means. Okay, this person will not carry the disease. There's no way this person can have a baby with Niemann-Pick disease, no matter who they marry. These two will be carriers. Okay, that's what it means by recessive disorder. So we can get some out of the way. So the absence of this enzyme causes the storage of large amounts of the sphingomyelin and cholesterol in the brain, bone marrow, liver, and spleen. Okay, so that's where this extra, because you're not going to break down the sphingolipids now. There's extra fat. So fat's going to go in the brain, which is going to disrupt the brain function, cause blindness, cause mental retardation, lack of development. It's going to get in the bone marrow. Bone marrow is responsible for your immune system, okay, and for erythropoietin, for establishing red blood cells. So you become anemic, and you have problems with your immune system. Liver filters toxins in the body. If all the fat deposits in the liver, liver can't function. And the spleen is the recycling for your red blood cells. Okay, you can live without a spleen. That's probably the least invasive one you can have. Okay, so the disease refers to a group of inherited disorders known as leukodystrophias, okay, or lipid storage disorders. The lipids that are made cannot be broken down. If it can't be broken down, they're going to end up somewhere. Okay, in different diseases, those lipids are stored in different areas. Okay, so the lipid storage disease in which certain fats accumulate in the tissues like the brain and the liver and cause damage. Okay, in Niemann-Pick's disease, the body is unable to break down fats completely, so they accumulate in the tissues and cause damage. Okay. So symptoms may begin when the baby is only a few months old, especially with Tay-Sachs disease. You will notice by the development of the baby. Parents may notice a delay in motor development, okay, or problems with feeding. Okay. Delay in motor development because it affects the myelin sheaths. Okay, it affects the brain. So the information not getting sent to the brain can't function correctly, so it can't send the right signals. So you have a delay in motor development. Infants may develop some motor skills. They quickly begin to regress. So at first they may develop some, and then they go back to back to the previous ways as they lose muscle strength and tone, as well as vision and hearing. Okay? So you start to lose your muscle mass. You lose the strength in your body. So you won't be able to start walking because they have no muscle strength. They lose the muscle tone. They start losing their eyesight and their hearing. Okay? And that's all because of these lipids being stored in the brain, causing interruption with the function. Okay. Disease progresses rapidly, and children typically die within the first few years of life. Okay? Most of the time, in Niemann-Pick's, Tay-Sachs disease, okay, children don't live beyond the first year or two. Symptoms of Niemann-Pick's disease. One of the difficulties in currently diagnosis of Niemann-Pick's disease is that its symptoms can be very different in each person. Because think about it. It's affecting the brain. It's not just playing a particular part in the brain. It's going in the brain, and then these fat cells are plugging out the brain. Depending where it plugs in the brain, you're going to get different symptoms. So not everybody's going to have the same symptoms. So it's kind of difficult to diagnose because you can't say this one set of symptoms applies to this disease and then this one, because everybody can be different when dealing with the brain. Does that make sense? Making sense? Because it's going to deposit in the brain in the fat, and depending where it's putting pressure on or what part of the brain it's destroying, depends what the symptoms are going to manifest as. So types A and C may have symptoms like, from Niemann-Pick's disease, an enlarged liver. Okay, one of the storage places is the liver. Liver gets extra fat in it, it's going to start getting enlarged, it's not function. Okay, brain damage, like we said, it starts in the brain, it's going to cause different areas. Difficulty walking and swallowing. They can't eat. Okay, again, depending what area of the brain is being affected, and then the coordination between walking. Okay, increased sensitivity to touch, become very sensitive. Okay, difficulty speaking, affects the speech center of the brain. Loss of muscle tone, hypotonia. Okay, so all those can affect the brain in different ways. Okay, it's not necessarily the same in each person. Okay, learning difficulties, that goes along with brain damage. Okay, you can have learning difficulties because certain parts of the brain can be affected. Treatment, what do we do for it? Not a whole lot. Okay, there are no specific treatments for any of the types of Niemann-Pick's disease or Tay-Sachs disease or any of the others. Okay, we have no specific treatments. We can help decrease the symptoms of the progression, but we can't treat that disease. Okay, bone marrow transplants have been attempted in several individuals with a type B. It's been attempted, hasn't been very successful. Okay. People with type C and D are often placed on a low cholesterol diet. Okay. Don't put in any additional, because the body makes certain fatty acids. Don't put any more fat than we have to. So we put a low cholesterol diet, so we're not adding to the lipids already there. Okay. Supportive care through nutrition, medication to treat symptoms, physical therapy are being followed by specialists who can help with the quality of life. Okay. But there is no specific magic bullet, this one. This is your bone marrow. This is your bone marrow on Niemann-Pick's disease. Okay. See this right here? These are fat cells full of triglycerides. Okay. You can see all these red blood cells here are being displaced. They're being pushed out. Okay. You're going to develop problems because the red blood cells are not going to be able to form like they should because these fat cells are blocking the way in the bone marrow. That's why they're trying bone marrow transplants. They clean this out. They put fresh bone marrow in without lipids, but the body still makes the lipids. It still makes the sphingomyelin. Okay. And it still has to deposit somewhere and it goes back into the bone marrow. So they haven't had success with bone marrow transplants because of that reason. This explains what we're always talking about as the genetics are infected. This is a recessive gene. So this is a carrier. John is a carrier, which means he's a carrier, means he has a big R and a little r. Okay, so what are the genes? The big R means it's a good gene. The little r means it's a bad gene. It lacks the enzyme. Okay, but the big R produces the enzyme. So John is good. Joan is unaffected. So Joan has two big R's of both her chromosomes, one from mother, one from her father, both produce the enzyme. So she's good. They have a baby. Okay, some of the baby will be unaffected, which means they'll get a big R from the John and a big R from the mom, Joan. And some will get a big R from the mother, and some will get a little r from John. Now that person is a carrier. And it's a 50-50 split. 50% will have no problem whatsoever, have not even the recessive gene. 50% will be a carrier for the recessive gene. Okay. Now, another couple down the street or across the world, okay, that has a carrier, which means big R, little r, carrier, big R, little r. They get married, Paul and Carol. They have babies. Okay. 50% are going to be carriers, which means they're going to have a big R and a little r. 25% will be affected, okay, which will mean two little r's, and then unaffected will be big R, little r. Okay. This child will probably die before being able to reproduce. Okay. This is an affected kid. That means the enzyme cannot be produced. Okay. Both genes are recessive now, lack the enzyme. He doesn't have a dominant gene that will have an enzyme to produce. So it's 25% of their offspring will be affected. Okay. Now, if Ian and Katie meet on the internet, okay, now they have babies. Okay. Both those are carriers. You have the same situation you did with Paul and Carol. The exact same thing. 50% are going to be carriers of their kids, 25% are going to be affected, and 25% are being unaffected. Remember, carrier means one gene has the enzyme, the other one doesn't. Affected means both genes do not have the enzyme, and unaffected means both genes have the enzyme needed to break down the phospholipids. That's how genetics works. That's a simplified version. So don't go to your genetics teacher and say, well, Dr. Goodall said. This is a simplified version of it. Right? This is how roughly it works. There's other variables involved. Okay, but this is the basics for how genetics works and how it's passed down from when there's a recessive gene disease. Does that make sense? Someone nod or give me a thumbs up or okay. I got a nod and a thumbs up.
Just to be clear, so in the tree that you make, like the genetic tree that you made right now,
right?
If there's a possibility for like 50, 25, 25, then it'll just be represented as three different kids.
No, not necessarily. And no, You can have 3 kids and they all can be unaffected. These are just odds.
Okay.
When these two get together, Katie and Ian get together, okay, they have a 50% chance of having a kid that's a carrier. When they have the next baby, they still have a 50% chance of having a kid that's a carrier. It's just the odds every time. It's not that they're gonna have 3 kids and one's gonna have each one of them. That's not
Okay,
what
so
that
they could
says.
have one kid and there's 25% chance that that child carries both recessive genes.
Bingo. That's
Okay.
it. That's exactly what it means. Okay.
Basically, it's the same thing as the
The
square.
odds are this. The odds say that. Right. They say the same thing with each kid. So if they have their 4th kid, that 4th kid has a 50% chance of being carried, 25% of being infected, 25% chance of being unaffected. Same with every kid, because it has that genes being transferred between the sperm and the egg when they fertilize.
All right.
Good point. Really good point. I'm glad you brought that up. All right. Any other questions on that? That's genetics 101 in a nutshell. Okay, Tay-Sachs disease, which is another lipid storage disease, okay? Caused by the absence of the enzyme hexose aminidase, which functions in the ganglioside metabolism. Okay, so again, you're missing the enzyme to break down the lipid. As a result of the enzyme deficiency, gangliosides, shown on the following slide, is going to accumulate in the cells of the brain, causing neurological deterioration. Okay, so with Tay-Sachs disease, the lipids occur in the brain, and the neurological portion of the brain, a lot of the brain starts to deteriorate from all the fat deposits that are in all the lipids. Okay? Those babies do not usually live that long. Like Niemann-Pick's disease, it's an autosomal recessive gene. Okay, so you have the same odds, same way, same genetic tree that we did as with Niemann-Pick's disease. Okay, becomes apparent in the first few months of life. Okay, so you won't, like someone just said, babies can be born. Some babies can be really fat when they're born. So you don't know if they're storing diseases or not, and their diet's not developed yet. But in the first few months, okay, the infant rapidly progresses to death within a few years. You get a decrease. I mean, they don't walk when they're supposed to. They don't develop. They don't show all the characteristic marks of development that an infant should show. Okay, you see that the first few months of life, and they don't last more than usually 1 to 2 years with Tay-Sachs disease. Okay, symptoms you can look for in the baby. You will see listlessness, irritability. No babies are ever irritable, right? Seizures, paralysis, loss of muscle tone and function. So the big key is the baby starts to develop muscle, and then they recess. The muscle tone gets less. You get blindness, you get deafness, delayed mental and social skills. Okay, all of those, okay, are areas where the brain is being destroyed. Okay, the brain's being deteriorated. That's what causes all of those symptoms right there. Okay, from the lipids being deposited in the brain. This is the MRI. This is a normal, I don't want to say normal. This is a, normal implies, this is a typical MRI of a brain. You can see all the grooves, the expansions, you can see all the, okay, all the dark area. Yeah, brains are, if you're looking at a brain under a dissection, they're gray and they're white. The gray area in the brain are your cell bodies. The white area is the axons that are sending information everywhere in the brain and the rest of the body. That's your myelin. Those are the myelin sheaths. Okay, so all those areas now are building up. So with Tay-Sachs, look at the difference. Look at all that is being condensed and compressed into each other. Okay, there is no more separation anymore. There's going to be no more brain function. That is slowly deteriorating the entire brain. Okay, you can easily see that on the MRI. Okay, another disease is Gaucher's disease. Gaucher's disease is more common, and you will see this in your clinics, okay, in your emergency room, things like that. Gaucher's is a recessive gene also, same genetic tree that we drew for the other ones, the same thing. Okay, results in deficiency of the enzyme glucose cerebroside. Okay, remember glucose cerebroside? Okay, normal situation, those breakdowns, glucose cerebroside. which is synthesis or degradation complex of glucose phenolides found in the cellular membranes. Okay. Remember, glucose riboside affects the myelin sheath, the single myelins. In Gaucher disease, glucose riboside builds up in macrophages, okay, found in the liver, the spleen, and the bone marrow. Okay. So Gaucher's disease, they build up, remember the glucose ribosides found in the cell membranes of macrophages, which destroy your invading pathogens into the body. Okay. So this glucose riboside builds up because the enzyme to break it down is not being transmitted in the disease. Okay. These cells become engorged with excessive lipid and displace healthy normal cells in the bone marrow. Okay. Just like the picture you showed before in the bone marrow. The symptoms of Gaucher's disease, okay, includes severe anemia. The red blood cells are being displaced in the bone marrow. So you're not going to make red blood cells at the right rate. Thrombocytopenia. Thrombocytopenia is a bleeding disorder. Okay, you reduce the number of platelets so your blood can't clot, like Van Willebrand's disease or hemophilia. Okay, examples of thrombocytopenia. And hepatosplenomegaly, which means an enlarged spleen and an enlarged liver. Hepato is the liver, spleno is the spleen, megaly means large. So enlargement of the spleen and liver. So with Gaucher's disease, okay, it's replacing the cell membrane, okay, in the macrophages and cause those to engorge. It's being stored in the bone marrow and causing that to fill up with fat instead of all the red blood cells. So it's causing anemia, thrombocytopenia. Okay, you're getting a very enlarged spleen and liver, those cells are being stored too, and they're just starting to expand. Okay, there can also be skeletal problems, bone deterioration, and secondary fractures. If you ever have bone deterioration, secondary fractures just mean the bone fractures in response to something that would not normally cause a fracture, like stepping off a curb can fracture a bone. Okay, here's a perfect picture of it. The one on the left, this is typical bone. This is normally what you'd see. You see the hips, you see how they come in, you see the pubic symphysis, you see good bone density, good support, you see the femurs coming down. This is Gaucher's disease. Okay, look at the difference. Look, here's the head of the femur bone. Look at the holes in it. Okay, look at the holes here. Look at the huge enlarged spleen right there. You can see right there. Okay, you can easily see the difference between those x-rays. The other one's patchy, full of holes, the bones aren't developed right. And it looks like these bones can shatter because they're hollow right here, which they are. Okay, so the pelvis that we're looking at shows a variation in bone density and deformities on both femurs, both the femoral heads. We saw that. In the pelvis, sclerosis extends in a broad swath on both sides from the acetabulum to the sacrum. Okay, those are the markings on the pelvis bone. On the right side, there's some flattening of the curves of the femoral head with osteoarthrosis and possible subarticular cyst. Okay, those are the patch areas that we saw. With the possible cyst means those dark areas. Remember, when you take an x-ray, if bone gets in the way, that x-ray beam bounces back towards you. So it leaves a white image on the x-ray. If the bone becomes patchy or eaten away, that x-ray beam goes through and starts to show a black pattern. So the black areas on x-ray is where the x-ray beam goes through. The white area is where it bounces back. It's not hitting the photographic paper. Okay? So the white area is where your bone is. All the little black areas or holes or the spot pattern is where the bone's being destroyed. So a cyst is usually a fluid-filled sac that dissolves the bone from an infection. Okay? And that's what you're seeing on those little areas. Okay? Irregular sclerosis is into the right femoral head, femoral neck of the femoral bone. On the left side, there's a possible producer acetabulate, which is a defect in the socket that causes the head of the femur to extend into the pelvis. You saw how that they didn't line up the same. That one side, that the head of the femur is well extended into the hip joint. Okay. So the articular surfaces are regular flat with collapse of the femoral head. So you see all this on the x-ray, okay? The bones are collapsing, they're porous, they're not healthy, they get secondary fractures on those. Broadening adjacent surface of the ilium and neck of the left femur. Again, just compare the x-rays what you're doing. There's sclerotic patches. that we saw, okay, from possible cyst. Diaphysis. The diaphysis is the junction of the long bones. Shows how the patch has medullary low density, and it thins the cortex. We have a cortex and medulla. That's we were talking about that in anatomy, not in this class. But the cortex is the outer portion of the bone, medulla is the inner portion. You see it thins the outer strength of the bone, so the bone can fracture very easily. And that one thing I pointed out, that big mass, that was the spleen. It's not supposed to be that big. Okay, here's the difference. Someone tell me what the healthy side is, left or right. That's the healthy side.
The left side?
That's what knees is supposed to look like. Okay, you got good bone support, good junction. You got your knee joint here with the femur coming down, okay, into the tibia and the fibula. Okay, you see the good extension, good socket here between those two bones. Here, look at the knee socket. It's completely destroyed. It's worn, almost worn down to nothing. Bones are flat. There's no articulation there. Your knees aren't going to work very well. And look at the difference, like right in here. Look at that. Can you see how white this whole area is on the left side and how patchy and dark it is on the right side? Those X-ray beams going right through that bone. There's nothing left. Okay, if you jump down a curve, you're going to fracture that bone. It's just nothing holding it. Same on both sides. You can see how it almost looks hollowed out on the X-ray, whether it's cysts or air sacs or just arteriosclerosis. Okay. Big difference in the X-rays with the Gaucher's disease. Disorder the metaphysis. Metaphysis is your growth plates on the bone. And adjacent diaphysis, that's the internal part of the long bone on the knee joint on both sides. That's the picture we just saw. The bone is widened, appears a modeling abnormality with the variable bone density. We showed you that in the different areas where bone was darker. There's some loss of bone in other areas where coarse strands of density in the diaphysis parallel the shaft. The whole length of that long bone, okay, was hollowed out. That's all that means. This appearance can apply some variation over time, okay? In the bone as a response to mechanical demands of weight bearing in the presence of random bone loss. So what's going to happen? You look at those two pictures, right? The healthy knee bone is going to withstand the force and the muscles of the knee. As that bone gets worse and worse from Gaucher's disease, it's going to give way. It's going to fracture. It's going to have to give. There's no strength, no support left on that bone. Okay? The cortex is irregularly thin. The cortex is the outer portion. That's what gives bones its strength. Inside the bone is your bone marrow. That's not where it gets strength. This is one of the treatments we do for Gaucher's disease. This is called enzyme replacement therapy, where we give the patient the enzyme that they're lacking in the disease so they can metabolize the liver. This is before they were given the enzyme. This is after. Look at the difference. And again, it's a fat storage disease, and you can see it. The spleen and the liver, that's where the spleen and liver located, right there. Okay? Look at the face. Okay? Look at the size of it. I mean, you can see the arms are roughly the same. Okay, fat storage doesn't occur in there, but you can see where the fat storage occurs in the spleen, in the liver, in the brain, okay, in the bone marrow. Okay, you can see the tremendous difference before and after enzyme replacement therapy.
What's the duration for that?
What's that?
What's the duration for that?
I'm not sure. They have to be. I know they have to be on the enzyme for life, but I don't know if with enzyme replacement therapy, I don't know if there's complications that still limit their lifespan. But I know it's a better quality of life, and they're not going to survive without enzyme replacement therapy. But anytime you start modifying enzymes in the body, something else usually happens. I'm not sure if they will live a normal life with that or not, but their quality of life will definitely increase with enzyme replacement therapy. Fabry's disease is an X-linked inherited disorder caused by deficiency of the enzyme galactosidase A. Okay, so it's linked to the X chromosome. So it's going to be more common in males. Okay, remember, females are XX, males are XY. So it's a disease of the X chromosome. If it's on the male, you only have one X chromosome. So you're screwed. Females have two. If it's on one X chromosome, they still got another one. It could be on that one. So the Fabry disease being much more common in males than females because it's an X-linked inherited disorder. Again, genetics 101. This disease afflicts as many as 50,000 people worldwide. Okay. Symptoms include pain in the fingers, toes, and a red rash around the waist. It's a very distinctive rash. It's not your, I'll show you a picture. It's not your regular rash that you see. If you see this in the clinic, you know one of your first things should be Fabry's disease. One of your first proposed diagnosis of it. And it begins to appear when the individuals reach their early 20s. So pain in their fingers and toes and a red rash around their waist. Okay. Patients are heat and cold intolerant, can have abnormal opacities of their eyes and the cornea. Okay. Raynaud's disease is one of the symptoms of this, which means you have cold intolerant. My daughter has Raynaud's disease. Her hands, if she's out in the snow, her hands turn blue. Her hands, she cannot handle the heat. She cannot handle the cold. Okay. She has to, if she is in the, when we were snowboarding, okay, she wears really thick, really good down gloves. Okay. So she can't handle heat and cold. They're intolerant to heat and cold. Okay. A preliminary diagnosis can be confirmed by determining the concentration of the enzyme alpha galactosidase A. Okay, so you look for those enzymes. Patients with Fabry's disease have increased risk of kidney and heart disease and a reduced life expectancy. Okay. This is an S-linked disorder, more common among males than females. Okay, that's the rash. Okay, that's not your typical rash. Okay? It occurs along the sides, along the back, and just in that area. It's kind of a deep purplish color, very distinctive for this disease. Okay, that's one of the aberrations of the cornea. If you look at a cornea, that's not supposed to be here. The funny thing is, it doesn't affect their sight. It doesn't have any effect on the sight. It's just an abnormality in the cornea that's visible when you look into the eye, but has no effect in terms of the focusing of the lens or anything else on how the eye works, which is really unusual. Okay, or you see patterns like that. That's a cornea. The whole thing's like a starburst pattern that goes across the cornea. And again, the eye still functions. It's just an aberration in the cornea from this disease, from the lipids being stored and during development of it. It's kind of cool. Steroids. Steroids are synthesized from a 5-carbon isoprene unit. Okay, now we have an isoprene unit. We've had glycerol units, we've had other units. Okay, now we have isoprene. We're going to build our steroids from. Steroids are 4 rings that are tied together. Okay, part of a diverse collection of lipids called isoprenoids and terpenes. Okay, those are your lipids when you deal with steroids. Your isoprenoids and your terpenes contain the steroid carbon skeleton. Okay, it's a collection of 4 fused carbon rings. And this is what they look like. This is the nucleus of a steroid. There's 4 fused carbon rings. There's 3 that have 6 carbons, and there's 1 that has 5 carbons. Okay, so here's a 6 carbon ring, 6 carbon ring, 6 carbon ring, 5 carbon ring. That's what the skeleton, that's what your steroids are based on. That's your isoprene unit, folds over, forms like a polymer, and forms a steroid nucleus. Okay, so your steroids are built upon this 4-carbon ring. Steroids are a member of a large, diverse collection of lipids called isoprenoids. Those are your isoprenes. Okay, all these components are built from 1 or more 5-carbon units called isoprene. So isoprene builds and folds into these ring structures to build your steroids. Okay. Terpene is a general term for lipids that are synthesized from isoprene units. That's what we call them, terpene. Examples of terpenes include steroids and bile salts. Okay, the lipid-soluble vitamins, chlorophyll, and certain plant hormones. So steroids, you know what steroids do? Build muscle mass in the human body or in other animals. They work, affects it very well for that. They also cause a lot of side effects of steroids. We had a Great Dane who I got him at 5 weeks old. His name was Apollo. We call him Apollo. And they were going to kill him because he had an autoimmune disease. He had he had a disease. He had to take steroids. We had to give him steroids and ketamine, which is basically a painkiller, since he was 5 weeks old. And he lived for 11 years, which is long for a Great Dane. This dog, he was 225 pounds when he died. He was a monster dog. Like I said, he was on steroids his whole life. So when he walked, I mean, you could see the muscles just bulge as he walked down the street. Everybody moved out of his way. No one walked in front of this dog. We put the kids in the stroller, the dog walked in front of him, everybody moved. It was amazing. His neck, we had to order a special dog collar. His neck measured 34 inches. Okay, that's the size of a bear, the head of a bear. That's bigger than my waist.
How tall is his back?
If he stood up, he was 9 feet.
Oh shit.
Yeah, he'd stand, put his paws, and he put his paws right on my shoulder, and his head would just tower above me. And he only listened to one person, and that was me. He listened to nobody else. I mean, he would go out in the backyard. This is when we live in Northern California, up in the mountains up there, and he would go out and lay with the deer out in the forest. And anybody could call that dog, he would not move. I would call him, he'd come right there. Great Danes only trained to one person. That's their breed. And he was a monster. And he was gentle, but you better not touch anything he's protecting. They're very territorial. We had a nursing friend over one night, and one of my sons was in his playpen. He was real little at the time, probably 2 years old or so. And my wife and I went in the kitchen to get something, and I just heard this big thud and a scream. She had bent down to pick up the baby when we weren't there. Nobody does that. And the dog didn't touch her, but he lunged and he put one paw on both sides of her head against the wall, and she wouldn't let her move. She never came back over again. He didn't hurt her at all, but he was right there. But yeah, he was on, and that's what steroids do to your body. I mean, he had muscles on top of muscles. Yeah, and steroids for 11 years. All right, so that's what steroids are. Bile salts, lipid soluble vitamins. Now, you've got to be careful with lipid soluble vitamins. You're dealing with patients. Vitamin C is water soluble. You cannot overdose on vitamin C. It's going to get, when you take too much vitamin C, what happens? What does your urine do? It turns yellow, right? Bright yellow. Take too much vitamin C, it just goes through your body and gets flushed out. Can't overdose on it. Lipid soluble vitamins, vitamin D, vitamin A, those you can overdose on and cause severe problems because they're stored in your fat. They're not going to go through your body. If you take too much of it, more than your recommended daily allowance, it's going to build up in your body and cause problems. So when you're doing nutrition with patients, there's a big difference. You can't just say vitamins. There's a big difference in handling lipid-soluble vitamins and water-soluble vitamins. Okay, because when you think about it, it makes sense. Water-soluble vitamins are just going to flow right out. Lipid-soluble are going to be stored in your body. They store in the lipids. Chlorophyll in plants for photosynthesis and other plant hormones. This is isoprene. This is a unit that builds up the basics that's going to build into all those ring structures and fold over. That's going to form the 4 carbon rings of your steroid backbone. That's isoprene. And it's just like a monomer. You get another polymer on it, because there's double bonds can open up and they can add to the next one. Okay. So we're talking about steroids. Steroids are like cholesterol. Cholesterol is in your cell membrane. It's a precursor to the bile salts released from your gallbladder. Male and female sex hormones, vitamin D, and adrenocortical hormones. Again, vitamin D is a lipid-soluble vitamin. Okay? You have to be very careful in your dosages of vitamin D. Okay, bile salts. Okay, bile salts build up; they can cause gallstones. Okay, can you take your gallbladder out and still survive?
Yes.
Yeah, because gallbladder does not produce bile. Bile is produced in the liver. The liver comes down, the excess is stored in the gallbladder. Okay, so if you have gallstones, things like that, you can take the gallbladder out. The liver is still going to produce bile. You're just going to lack the storage facility now. So you can't have as many fatty foods that you used to if you have your gallbladder removed. But the gallbladder does not, it's a big misconception. A lot of patients will tell you, oh, I have my gallbladder out, so I don't produce bile anymore. The liver produces bile. Okay? If it backs up, it goes into the gallbladder for storage to release later. So you get more of an immediate response and more bile salts released into the intestine when you need it. Okay? Okay? So the gallbladder itself does not produce bile. It's done by the liver. Okay? Male and female sex hormones and adrenocortical hormones. Okay? They have been linked to atherosclerosis. Atherosclerosis is hardening of the arteries. Okay? When you get too much, things build up, the blood becomes thicker and starts to harden up in the arteries and starts to thin the lumen of those blood vessels, which can cause cardiovascular disease, can cause stroke, all kinds of nasty things. Okay? And from the liver. I was just about to say where are we? Oh, did lose its connection? Okay, so bile salts, like we talked about that, being stored in the gallbladder and coming from the liver. Okay, they're important in lipid digestion. They break down fats. So like I said, in your patients that have had gallbladder surgery, they have to modify their diet. They still will produce bile, the bile salts to help break down lipids in their intestines during their diet, but they won't have as much as a person that still has a gallbladder intact. Okay, because that storage facility is now gone. Cholesterol is a common steroid. Okay, it's found in the membranes of almost all animal cells. Okay, it's amphipathic molecule. What's amphipathic mean? It is polar end and a non-polar end. So it can emulsify things and is readily soluble in a hydrophobic region of membranes. So it's amphipathic cholesterol. The hydrophobic or the fatty chain region dissolves in the middle of the membrane, which is your hydrophobic region of the membrane. And then the polar end sticks out to where the extracellular fluid is or the cytoplasm of the cell on the inside. Okay. It is involved in regulation of the fluidity of the membrane as a result of the non-polar fused rings. Okay, so those non-polar rings are fused in that steroid structure, those 4 rings we talked about. So it can go into the membrane and can move in the membrane. It allows it to float through the membrane. Remember, cell membranes are completely fluid. They don't hold a shape. They move, they bend, they swell, and this cholesterol can go with it because the non-polar, the hydrophobic end, is dissolved inside the fatty acid chains of the lipids that form the membrane wall, the cell membrane. So it can move right along with it. The hydroxyl groups are polar, okay, and function like polar heads of the sphingolipids and the phospholipids, just like the other ones, okay? They allow that end to bond with the water or the extracellular fluid or in the cytoplasm of the cell. So there's a strong correlation between the concentration of cholesterol found in the blood plasma and heart disease, particularly atherosclerosis, hardening of the arteries. Okay, so that's why we check for cholesterol. I think if you have a cholesterol below 150, you have like minimal chance of a heart attack, heart disease. As cholesterol builds up in your blood plasma, okay, it causes plaque to be deposited on your artery. So if this is your artery, okay, blood flows through here, all your red blood cells. Actually, let me use red for red blood cells. That makes it more sense. Okay, and they flow through your arteries. That goes to your brain, to your heart, stuff like that. Now, as cholesterol molecules, let's use green for cholesterol. As these molecules of cholesterol, remember, they have polar ends and non-polar ends. So the polar ends are going to dissolve in the blood plasma, which is 99.9%, was 99% water. Okay. As it goes through the water, it has a non-polar end, and that's going to stick to the non-polar ends of the cell membranes along that lumen of the blood vessel. So as your blood vessel goes, this is your blood vessel, and now the cholesterol is starting to stick to the non-polar areas. Okay, so what it's done now is decrease the size of that blood vessel. So very few red blood cells can get through now. And this is called plaque, and that's called atherosclerosis. So when you have patients with that, they're very good subjects for stroke, if this occurs in the brain, if the artery gets clogged up in the brain, you get a stroke. Okay, it cuts off the blood supply. If that artery is going to the heart, you get a heart attack because it cuts off the blood of those supplies. Okay, they put in stents. They do still that for surgery to help open those areas back up with atherosclerosis. Okay, but that's what causes it with cholesterol because it's amphipathic. It bonds into the membranes and it builds up this plaque on the wall and decreases that lumen of the blood cells smaller, smaller, smaller till it shuts it off. If it shuts it off in the brain, you got a stroke. Shuts it off in the heart, you have a heart attack. Okay. So cholesterol in combination with other substances contributes to narrowing of the artery passageway. So you narrow that passageway in the artery. That's what causes the problem. As narrowing increases, more pressure is necessary to ensure adequate blood supply. High blood pressure develops. You also develop hypertension because now you've got to force blood through a smaller gap. You got to try to force just as much blood through a smaller gap, which means you've got to push it harder, which means your blood pressure is going to go up. So not only does it cause cardiac arteries, decreasing the size of the lumen, potential stroke or heart attack, now it's also increasing your blood pressure, which causes other problems. Increased blood pressure causes damage to your kidneys, damage to your liver, because it can't handle that extra force in those organs. Okay? So you got all kinds of problems with too much cholesterol in your diet. So hypertension is also linked to heart disease. Okay? Causes the heart to pound a lot more, which wears it out. Okay? Bile salts. The bile salts produced by the liver stored in the gallbladder are emulsifying agents. They're amphipathic. They dissolve the lipids that is in your diet in your small intestine. Okay? The liver goes down the common hepatic duct, which feeds into the duodenum, which is the beginning of your small intestine. That's where all your resorption occurs. Okay? So the bile salts go into the duodenum of your small intestine, and that's where you get emulsification. That's where you get your breakdown and dissolving of the lipids in your diet. Okay? The polar hydroxyl groups interact with water. Remember, it's amphipathic. There's your polar, and the hydrophobic regions bind to the lipid. So they bind to it and then carry it to where they need it to dissolve it, break it down. So following a meal, bile flows from the gallbladder to the duodenum, which is the region of the small intestine. Okay. Okay. Remember, bile is stored in the gallbladder. It's also released from the liver. Okay. So if the gallbladder is missing, it comes from the liver and goes down, but just not as much. Here, the bile salts emulsify dietary fats into small droplets. Remember, they break them down. They like micelles. They form those groups around them, being readily digested by lipases. Lipases are enzymes that break down lipids. Okay, those are also found in the small intestine. Steroids also play a role in the reproductive cycle. Okay, in a series of chemical reactions, cholesterol is converted to the steroid progesterone. Progesterone is used during pregnancy to allow a fertilized egg to implant into the uterus. Okay, without progesterone being produced, the fertilized egg will not implant and pass right through. Okay, it causes a thickening of the endometrial lining. Okay, and it causes a preparation for the egg to implant there when it passes through there. Okay, it's produced in the ovaries and in the placenta. Progesterone is responsible for both the successful initiation and then the completion of the pregnancy. So it initiates it by allowing implantation and then keeps the development of the placenta so the fetus can grow and get nourishment. It prepares the lining of the uterus to accept the fertilized egg. Okay. Fertilization does not occur in the uterus. Okay. Fertilization occurs in the uterine tubes up by the ovaries. That's where fertilization occurs. And the egg starts to expand, multiply over and over and over again. Okay. Releases progesterone, gets the uterus ready. So when it comes down 3 or 4 days later towards the uterus, then it can bind into the wall. Okay. Once the egg is attached, progesterone is involved in the development of the fetus and plays a role in the suppression of further ovulation during pregnancy. So it stops you from having more eggs being ovulated. Prepares the lining of the uterus with the fertilizing egg as it passes down from the ovary into the fallopian tubes, into the uterus. It gets stopped in the uterus and embeds in the endometrium because of progesterone. Without progesterone being there, it wouldn't be able to embed in the wall. Once the egg is attached, progesterone then stops more eggs from being released from the ovary. So it stops further ovulation during pregnancy. That's why you don't have a period during pregnancy, is because of progesterone being produced. And newer birth control formulations include both progesterone and estrogen. So the new ones contain both steroids. Okay, testosterone. Testosterone is the male sex hormone found in the testes. Okay, and estrone, a female sex hormone, estrogen and estrone, are produced by chemical modifications of progesterone. Right. These hormones are all the development of male and female secondary sexual characteristics. Okay. Many steroids, like progesterone, play important roles in development of birth control agents because it stops you from ovulating. 19, norprogesterone was one of the first synthetic birth control agents. That was one of the first ones produced. It is approximately 10 times as effective as progesterone in providing birth control. As far as the synthetic one. However, its use was limited because it could not be administered orally. It had to be given by injection. Okay, so it kind of limited where it could be used under what circumstances. Okay. But a related compound, Norludin, which has a chemical name 17-alpha ethinyl 19 noresterone, was found to provide both strength and effectiveness as the same as a 19 progesterone. This one can be taken orally. So that's the most commonly used now, is that combination pill. Currently, combination contraceptives are prescribed most frequency because you have both steroids working together to prevent pregnancy. These include a progesterone and an estrogen, because the combination of the two is the most effective. These newer products confer better contraceptive protection than either agent that is administered individually by itself. They're also used to regulate menstruation in patients with a very heavy menstrual cycle. First investigated in the late 1950s and approved by the FDA in 1961, there are at least 30 different combination pills that are available on the market today as birth control pills. In addition, a transdermal patch for the treatment of postmenopausal osteoporosis is being investigated. Okay, at the end of menopause, a lot of women don't produce estrogen anymore. The bones start to become very thin, and you get what's called osteoporosis, which actually the bones become just like in Graves' disease. Okay, bones become hollowed out, they become very narrow out and minded, and you can break bones very easily. And one of the treatments using is a transdermal patch right now, which has estrogen in it to help prevent this osteoporosis postmenopausally. Okay, all these compounds, how do they work? They trick the body into thinking it's pregnant. If the body thinks it's pregnant, it doesn't release another egg. That's how it works. Okay, they just make the body think it's pregnant. When oral contraception is discontinued, ovulation usually returns, and it takes about 3 to 4 menstrual cycles for it to start kicking back in again. So you don't just stop taking the pill on Monday and try to get pregnant on Tuesday. It doesn't work that way. It takes about 3 to 4 months for the cycles to continue back into a normal rhythm to start ovulating again. Although there have been problems associated with the pill, okay, it appears to be an effective, safe method of family planning for much of the population. Part of the problem with the pill was thrombocytosis in females that are susceptible to certain types of heart disease to start with. Cortisone is also important to the proper regulation number of biochemical processes. We use cortisone in our body a lot. For example, it's involved in the metabolism of our carbohydrates. When we break them down, we use cortisone. Cortisone is also used in the treatment of rheumatoid arthritis, used for asthma, gastrointestinal disorders, and many skin conditions, and a variety of other diseases. We do cortisone injections for pain, for ligament tears, okay, painful joints. We do cortisone injections in there. Okay. Treatment with cortisone is not without risk. Okay. Some of the possible side effects of cortisone therapy include fluid retention, sodium retention. Now, you got to remember that those go hand in hand. Remember, salt is a polar material. Salt dissolves in water. So when you retain water, you retain the salt with it. to retain sodium. Potassium loss, that can lead to congestive heart failure. Potassium levels have to be monitored carefully. If you lose too much sodium, your heart will not get the signal to beat correctly. And you can call it congestive heart failure. Other side effects could be muscle weakness, osteoporosis, gastrointestinal upsets like peptic ulcers, neurological symptoms like vertigo, headaches, convulsions. Okay. So it's not without its side effects or with risk of side effects. Okay. Aldosterone. Aldosterone is a steroid hormone produced by the adrenal cortex, secreted in the bloodstream when blood sodium ion levels are low. Aldosterone is the same thing as anti-diuretic hormone. Okay, it keeps the body from being dehydrated. Okay, when sodium levels are low, how do we get sodium levels back up? We reabsorb sodium from our intestines. Okay, so aldosterone causes sodium to come back up, which is water also. When sodium moves with the water, so when you reabsorb water, you reabsorb sodium, and it prevents the kidneys from filtering out water, okay, and sodium, so it stays more in the body. That's why it's called an anti-diuretic hormone. It's the opposite of being on a diuretic. Okay, upon reaching its target tissue in the kidney, aldosterone activates a set of reactions that cause sodium ions and water to be returned to the blood. Okay, like I said, sodium and water go together, so we bring it back from the intestines after it's been digested. Okay, put it back to the bloodstream. We go to the kidney. We stop it from being excreted in the first place, so it doesn't filter sodium. So we retain sodium and water through the kidneys. If sodium levels are elevated, aldosterone is not secreted from the adrenal cortex because you want the exact opposite to occur. And the sodium ions filtered out of the body by the kidney will be excreted. They don't get reabsorbed then because the levels are high enough. Steroid examples. Again, here's your 4-membered ring. 3 6-carbon rings, 1 5-carbon ring. Okay, that's your steroid background. And this is cholesterol. You can see the long chain fatty acid, methyl group, and then your hydroxyl group. That is cholesterol for steroid. This is testosterone. Okay, again, same steroid background. Same steroid blueprint. Okay, 1, 2, 3. There's your 4-ring steroid. 3 6-carbon rings, 1 5-carbon ring, and that is testosterone. That's progesterone, the female sex hormone. Again, same steroid backbone. Backbone's what we're looking for. Okay, cortisone, same steroid background. Hydrophobic and hydrophilic regions. These are your hydrophilic regions. Hydrophobic is all that long with carbons. Okay, and adrenal corticoids have 2 groups, these steroid hormones. Okay, adrenal corticoids can be mineral corticoids, can't talk right now. Mineral corticoids, which regulate your iron concentration, your sodium and potassium. Okay, and glucocorticoids, which enhance carbohydrate metabolism, regulating glucose. Okay, those are 2 types of adrenal cortical groupings we have. Or steroid hormones. Cortisol increases glucose and glycogen in the body. So it increases the amount of glucose, and then the glycogen is a storage form of glucose. Okay. Along with its ketone derivative, cortisone are anti-inflammatories. So cortisol and cortisone are anti-inflammatories. That's why you inject it into a joint that's inflamed. You got an elbow or knee joint that's hurting with inflammation there, they do a cortisol injection in there. Okay, they put its cortisone in them. Okay, this is a very, very painful injection because they take the cortisone, put it in, and they move the needle around until they find the area which is the most inflamed, which is the most painful, and that's where they inject it. Okay. Another derivative is prednisolone. Prednisolone is used to treat asthma and inflammation. Steroids and the treatment of heart disease. We use steroids to treat heart disease. The foxglove plant. Foxglove plant is Digitalis purpura. It's a herb, produces one of the most powerful known stimulants of the heart muscle. So Digitalis is a stimulant of heart muscle. and it's found in nature, the foxglove plant. The active ingredients of the foxglove plant, digitalis, are the so-called cardiac glycosides or cardiotonic steroids. Includes digitoxin, digoxin, and gitelin. That's what the foxglove plant looks like in nature. Okay, this is digitoxin, a very powerful heart stimulant. Okay, whoops, I backed it too far. Okay, that's digitoxin. You can see what it looks like. You can see this. Here's your steroid backbone, your 5 carbon rings, and then other rings attached to it. These drugs are used clinically in the treatment of congestive heart failure, which results in the heart is not beating with strong, efficient strokes. Patients with congestive heart failure are going to come in, they're going to be short of breath, they're going to be out of breath, they're going to have a low pulse oximeter for your partial pressure of oxygen, usually below 90, and they're going to have swelling, especially in their legs, most of the time the lower extremities in their legs, okay? Because the blood cannot be returned back to the heart. The heart's not beating strong enough. So those are your signs of congestive heart failure, where the heart is starting to beat less efficiently. When the blood is not propelled through the cardiovascular system efficiently, fluid builds up in the lungs and lower extremities. Like I said, you have swelling in the legs, and you have in the lungs where they're out of breath. They come in the clinic and they can't catch their breath because now there's lungs, there's water, okay, built up, or fluid built up in their lungs. Okay, the major symptoms of congestive heart failure are an enlarged heart. The heart tries to be stronger and stronger. It just gets larger and larger to try to compensate for it. You have weakness, edema, which is swelling, shortness of breath, fluid accumulation in the lungs, making it very difficult to breathe. Those are all the major symptoms of congestive heart failure. Okay, this condition was really described in 1785 by William Withering, found a peasant woman whose folk medicine was famous as a treatment for chronic heart problems. She was using foxglove. Okay, her portion contained a mixture of more than 20 herbs, but Dr. Withering, a botanist as well as a physician, quickly discovered that foxglove was the active ingredient in the mixture. That's what was causing the success in getting the heart to beat stronger and prolonging a patient's life with congestive heart failure. Okay, he used digitalis purpura successfully to treat congestive heart failure and even described some cautions in its use. Okay. The cardiotonic steroids are extremely strong heart stimulants. A dose as low as 1 milligram, that's a very tiny amount, okay, increases the stroke volume of the heart and increases the strength of the contraction, and it reduces your heart rate because more blood is pumping more efficiently now. It doesn't have to beat as many times. So it increases the volume, how much blood is moved per contraction, and it increases how strong the heart contracts, how much it squeezes blood out of it. And then it slows the heart rate down. That's what digitalis does. When the heart is pumping more efficiently, because of stimulation by digitalis, the edema disappears. You got liquid there built up like in the legs. As the blood's flowing more freely and it goes through the area, now it can return that fluid in the extremities back into the venous system and go back into the heart. Okay. Digitalis can be used to control congestive heart failure, but the dose must be determined and monitored because the therapeutic dose is close to the dose that causes toxicity. Okay. The heart can beat so fast that it destroys itself. So you have to be very careful with digitalis, okay? Because it's a very powerful drug. Symptoms that evolve from high blood levels of cardiotonic steroids, okay, like digitalis, include vomiting, blurred vision, lightheadedness, increased water loss, convulsions, and death. Okay, so you have to be very careful in utilizing digitalis with your patients. Okay. Only a physician, cardiologist, someone else that's using it can determine the initial dose and maintenance schedule for an individual to control congestive heart failure and avoid the toxic side effects. There you go. All right. Draw the structure of the steroid nucleus. Note the location of A, B, and C, and D rings. What did we say the nucleus was? We said it was 4 ring, carbon rings, right? We have a 6-member ring. We have a 6-member ring. We have a 6-member ring. And we have a 5-member ring. That's our basic structure of steroids. A, B, C, and D. That's a better picture than I can draw. Okay, A, B, C, and D. That's your steroid nucleus. 3 6-carbon rings, 1 5-carbon ring. Okay. 17 total carbons. What is meant by the term fused rings? It's where a ring comes together and share carbon atoms and share a bond. Okay, 2 rings are joined together, they share a bond at 2 points. This point here and this point here, and they share this bond between 2 carbon atoms. Okay, that's one side of the A ring and one side of the B ring. Okay. Any questions so far? We covered a lot of that. All right. Waxes. Waxes are also esters, just like fats were. Kind of the same properties here. We use 1 alcohol instead of glycerol. So you don't use glycerol for waxes. You use alcohol. But the esters are the same way. Esters are long chain fatty acids. Okay. They are long chain alcohols. Okay. So instead of having a short chain like glycerol, now we've got 2 long chains, the carbonyl group and the oxygen group. So the longer the chains, the higher the melting point. Why? Because the longer the chains, the more they can stick together and increase their London dispersion forces. Okay. And they'll stick together more and be harder to pull apart. That's why they're waxes. That's why that liquid's at room temperature, but they're waxes at room temperature. Protects the skin of plants and the fur of animals. Okay. Examples of waxes includes carnauba from the Brazilian wax palm. Beeswax. Okay. So waxes are derived from many different sources, have different chemical compositions depending on what they're derived from. Okay. Paraffin wax, like used with candles. Okay. It's composed of solid hydrocarbons, usually straight chain compounds. Natural waxes, generally composed of long chain fatty acids, esterified to long chain alcohols. So now we're taking our long chain fatty acid and our long chain alcohol, hydration reaction, removing water between the two and creating an ester. And then we have a long chain fatty acid component and a long chain alcohol component. Because the long hydrocarbon tails are extremely hydrophobic, remember hydrocarbons are nonpolar. Okay, waxes are completely insoluble in water. You take beeswax and put it in water, it's not going to dissolve in there. Okay, it's hydrophobic. You have 2 extremely long hydrocarbon chains. So waxes are solid room temperature, just like animal fats, and it's due to their high molecular weight, just like with animal fats. Okay, the longer the carbon hydrogen chain, the more solid it's going to be room temperature, the less solid it's going to be in water. And the more it's going to stick together, making it a solid at room temperature. 2 examples of waxes. Okay, our miracle palminate was a major source of beeswax and whale oil, spermatocytic wax from the head of the sperm whale, which is composed of cetyl palmitate. Okay. So this is beeswax, and you can see this is the carboxylic acid side. Okay, and this is 16 carbon, sorry, it's using an even number. Okay, and this is the alcohol side. Okay, this is 30 carbon atoms. So you can see a lot more carbon atoms than we're used to dealing with the triglycerides. So this is a long chain alcohol and a long chain carboxylic acid. And that results in an ester, which results in a wax. And this one is beeswax, and this one is whale oil. Okay. Again, long chains on both sides of the carbonyl group. So natural occurring waxes, we have a variety of uses for them. Lanolin, it's a protective coating for hair and skin, and we use it we produce skin creams and ointments. And we use lanolin. Carnauba wax is used in automobile polish. Okay. Whale oil, once used as fuel in ointments and in candles. Okay, synthetic waxes have replaced whale oil due to a large extent because of the efforts to ban the hunting of whales. So we started making the wax instead of using it out of whales. Complex lipids. Complex lipids are those bonded to other types of molecules. So if a lipid and you bond to something else, that's a complex lipid. You have lipoproteins. Lipids are bonded to proteins. Molecular complexes are found in the blood plasma. They contain neutral lipid core of cholesterol esters and other triglycerides, triacetylglycerides, TAGs. Okay? So it's neutral lipid core of cholesterol esters, and the other triglycerides. Surrounded by a layer of phospholipids. That's on the outside because it's polar. And cholesterol, that's your cholesterol ring. That is non-polar, it's hydrophobic. And protein, that's what makes it a protein, a lipoprotein. There's different classes of these lipoproteins. There's chylomicrons, there's VLDL, there's LDL. I hit the wrong button, that's why it's not doing it. And there's HDL. Okay. The chylomicrons and the VDLL stands for very low density lipoprotein. LDL stands for low density lipoprotein. HDL stands for high density lipoprotein. Okay. Now, these complex lipids are lipids that are bound to other types of molecules, like we said, with protein. The most common important complex lipids are the plasma lipoproteins. They're responsible for transport of other lipids in the body as it transfers them through the bloodstream. Okay. Lipids are only sparingly soluble in water. They're not very soluble in water. Movement of lipids from one organ to another goes through the bloodstream, requires a transport system. That transport system is what we use is plasma lipoproteins that occur in the blood plasma. Okay. Lipoprotein particles consist of a core of hydrophobic lipids surrounded by amphiperic proteins, phospholipids, and cholesterol. Okay. So the hydrophobic is going to attract to the non-polar amphipathic part. Phospholipid is going to be the polar, and then the cholesterol is non-polar. Okay. And this is how they form. Okay. This is a plasma. Oops, I dropped my pen. Where did it go? It's way down there. Oh, I'll get my pen back here. Okay, those are a model of the structure of the plasma lipoprotein. Okay. Lipoprotein composes a shell of protein. Okay. And then cholesterol and phospholipids surrounding many hydrophobic molecules, like your triglycerides and cholesterols are inside. So this forms your bilayer. These are your long chain fatty acids you can see here. And this is your phosphate heads. Okay. So this is polar on the outside. This is non-polar on the inside. Polar is hydrophilic, non-polar is hydrophobic. So other molecules that are also nonpolar can exist inside the cell, like triglycerides. Triglycerides are nonpolar. Cholesterol is nonpolar. Okay, you have long chain fatty acids, long chain alcohols on them. Okay, so that's how that gets rearranged. And you have your proteins get wedged inside this membrane to help for structural support in the membrane. Okay, so chylomicrons. Chylomicrons are very large and very low density, less than 0.95 grams per milliliter. They transport triglycerides from your intestine to adipose tissue, to your fat tissues. That's where it moves it from. Very low density lipoproteins, VLDL, are made in the liver, and they have a density of 0.95 to 1.019. They transport glycerides from liver to tissues. Okay, so the chylomicrons go from your intestines to your adipose sites. Your very low density lipoproteins go from your liver to other tissues of the body. Low density lipoproteins carry cholesterol to tissues. Their density is about 1.019 to 1.063, so much more dense than VLDL. And then high density lipoproteins. Those are made in the liver, and those have the highest density. Okay, those are they scavenge excess cholesterol, esters in the body. Okay, that's what they do. They pick up on it. Okay, this is your good cholesterol, your high density lipoproteins. Okay, these bind to cholesterol esters that it finds in the body and transports it out of your bloodstream into your liver. Remember what cholesterol does in your bloodstream? Okay, it binds to the cell walls where there's hydrophobic areas and starts narrowing your blood vessels, causing atherosclerosis. Okay, so the good cholesterol is HDL. Okay, what that does, it goes through the body looking for the bad cholesterol and transports it back to the liver, getting it out of the bloodstream. So chylomicrons go from the intestines to adipose cells. Very low density lipoproteins go from the liver to other tissues. LDL does the same thing. It carries cholesterol to different tissues. And HDL, the high density lipoproteins, are your good cholesterol that binds cholesterol in the blood and transfers it back into the liver. Okay, and this is what they look like. These are your chylomicrons. Okay, these are your VLDL, very low density lipoproteins, low density lipoproteins, and high density lipoproteins. You can see the protein. There's only one here, there's 3 here, there's 4 here, and there's 5 here. So you see the amount of protein in the membranes increases as it goes up. Okay, so you get your very low density lipoproteins have very little protein. Your low density lipoprotein has just a little bit more, and your high density lipoprotein has a lot more protein in the cell membranes. Okay? So if you look at the cholesterol content, what they're looking at, in your chylomicrons, your cholesterol is 5%. In your very low density lipoproteins, the cholesterol level is 14%. Okay? In your low density lipoproteins, cholesterol level is 45%. And in the high density lipoproteins, your cholesterol level is 20%. Okay? So it's just an effective way of transporting cholesterol out of the bloodstream for the high density lipoproteins. Okay? But the big thing is the protein amount. If you look at the proteins make up 1% in the chylomicrons in the cell membrane. Proteins make up 8% in the very low density lipoproteins. Proteins make up 25% in the low density lipoproteins. Okay? And in the high density lipoproteins, protein makes up 45% of that cell membrane. Okay? Much more protein for the good cholesterol than all the other ones. So chylomicrons are aggregates, conglomerations, or groupings of triglycerides. Okay, the triglycerides that form together, and proteins that transport delivery triglycerides to cells throughout the body. Okay, so the chylomicrons are triglycerides are proteins that stick together, and they transport what you've eaten there, triglycerides, to cells throughout your body. Okay, not all lipids in the blood are derived directly from the diet. Okay. Triglycerides and cholesterol are also synthesized in the liver. So we make our own cholesterol and triglycerides. Okay. And are also transported through the blood in lipoprotein packages that are made. Triglycerides are assembled into very low density lipoprotein particles that carry the energy-rich lipid molecules to tissues requiring energy or to adipose tissues for storage. So triglycerides is how we store our glucose, how we store our molecules, right? We store energy, our phosphate bonds. Okay, they go to the very low density lipoproteins. Okay, they carry these to other tissues that need energy. And they can deposit our triglycerides there because that is our energy source. Okay, we break those apart to form an energy back again. Or if we don't need the energy, then it goes into fat tissue where we have it for storage so we can break it down later. So similarly, cholesterol is assembled into low density lipoprotein particles for transfer from the liver to peripheral tissues. Well, the entry of the low density lipoprotein particles into the cell depends on specific recognition and the binding between the low density lipoprotein and a protein receptor within the membrane. Okay? So the cells has to recognize the low density lipoprotein and bind to it so the cholesterol can be delivered to it. So low density lipoprotein receptors are found in the membranes of cells outside the liver and responsible for the uptake of cholesterol by the various cells, by the various tissues. You have the receptors for them to be not modified that they can deposit that there. So low density lipoproteins, okay? The lipoproteins bound to cholesterol bind specifically to the low density lipoprotein receptor, and the compound is taken into the cell by a process called receptor mediated endocytosis. So it binds the receptor, it keys it to go, okay? And you get endocytosis. Remember endocytosis is when it hits it comes, here's your cell membrane and here's your cholesterol. What they do is they bind to these receptors here and it triggers the membrane to start to cave in. And then as it caves in even more, it starts to seal itself off. And that goes inside the cell. Okay. That's receptor-mediated endocytosis. So the membrane begins to pull in the cell at the site of the low-density receptor complexes. This draws the entire low-density particle into the cell. Remember, endocytosis incorporates the whole thing and brings it in. Eventually, the portion of the membrane surrounding the LDL lipoprotein pinches away from the cell membrane and forms a membrane around the LDL particles. Okay, so these membranes are constantly changing. Okay, they're fluid and they readily flow. They can make and form a vesicle or endosome containing the lipoprotein particles. Okay, so it has to activate the membrane receptors. The LDL receptors, low density lipoprotein receptors, were discovered in investigation of familiar hypercholesterolemia. That's where your diet does not include cholesterol, but your body still makes a lot of cholesterol. So when a cell needs cholesterol, it synthesizes the receptor, migrates to a coated region of the membrane. The captured cholesterol is absorbed by endocytosis, grouping it in as one thing and fusing the membrane. Failure to make the receptor is the most common problem encountered. If you don't make the receptor, the cell does not absorb the cholesterol and the cholesterol builds up in your bloodstream. That is hypercholesterolemia. Hypocholesterolemia. Okay? It means you're not making the receptor, so when the triglycerides are bounded to the LDL, low density lipoproteins, and it tries to trigger the receptor, they're not there, so you don't get endocytosis and it stays in the bloodstream. Okay? This is endocytosis. These are your receptors being activated here. Endocytosis is your cell membrane coming inside the cell. This is all the inside. The rest is outside the cell. Okay? And then as that memory goes along, it starts to come back up and eventually pinches off and it forms a micelle inside the cell. Okay? So what's activated is these low density liporeceptors. That's what gets activated. That causes the endocytosis. If those receptors aren't there, then you don't get endocytosis and you don't get absorption of cholesterol into the cell. Okay? And that's what causes the disease site hypercholesteremia. Okay? So LDL, low density lipoprotein, binds to the LDL receptors, causes endocytosis, and it pinches off and moves them inside the cell. So cellular digestive organs known as lysosomes. We talked about lysosomes, okay? And they fuse with endosomes. Lysosomes contain materials that break down peroxide, break down other types of cell, other organ, whatever, infuse in it, and it's going to break down the cholesterol. Okay? The fusion is accomplished in the membrane of the endosome and the lysosome flow together to create one large membrane-bound vesicle. So the lysosome and the endosome fuse with the endocytosis, what was formed, and create one large membrane-bound vesicle. Hydrolytic enzymes from the lysosome digest the entire complex, release cholesterol into the cytoplasm of the cell. So it breaks it apart, releasing the cholesterol. So their cholesterol inhibits its own biosynthesis and activates an enzyme that stores cholesterol in cholesterol ester droplets. So once cholesterol is produced in the cell, it activates another enzyme that stores the cholesterol and stops the production of more cholesterol. Okay? So the body stops making it. High concentrations of cholesterol inside the cell also inhibit the synthesis of low density lipoprotein receptors, because you're not going to make as much there, to ensure that a cell will not take up too much more cholesterol. So once it's had enough cholesterol, it starts making the receptors, so you don't get more endocytosis of the extra cholesterol in the bloodstream. So people with a genetic disorder and the gene coding for the low density lipoproteins, receptors do not take up as much cholesterol, so more of it stays in the blood. Okay? As a result, they accumulate LDL cholesterol in the plasma. This excess plasma cholesterol is then deposited on the artery walls, like we talked about, causing atherosclerosis, narrowing of those blood vessels, which can cause a stroke if it's in the brain, or heart attack if those vessels lead to the heart, to provide oxygen to the heart. The disease you talked about is called hypercholesterolemia, okay, that lacks LDL receptors. It fails to make LDL receptors. So the cholesterol cannot be incorporated into the cells and stays in the bloodstream. Liver lipoprotein receptors enlarge large amounts of cholesterol to be removed from the blood, ensuring low concentrations of cholesterol in the blood plasma. Okay, so liver protein, just like the other cells. So everything being equal, the person with the most lipoprotein receptors will be the least vulnerable to high cholesterol diet and have the least likelihood of developing atherosclerosis, because the cholesterol is being absorbed out of the bloodstream by the low density lipoprotein receptors on either the liver or the other cells. There's also evidence that high levels of HDL, that's your high density lipoprotein, in the blood help reduce the incidence of atherosclerosis, perhaps because HDL carries cholesterol from the peripheral tissue back to the liver. So it takes it out of the bloodstream. Remember, HDL is your good cholesterol. So in the liver, some of the cholesterol is used for biosynthesis to create bile, secreted in the intestines, which is then excreted. So secreted in the intestines used to break down fats in the duodenum, and then excreted from the body. A final correlation has been made between diet and atherosclerosis. Okay? People whose diet is high in saturated fats tend to have high levels of cholesterol. Okay? Remember, what is saturated fats? No carbon-carbon double bonds, all single bonds. Has the most hydrogen you can possibly have on the molecule, which means it's going to stick together and be a solid at room temperature rather than a liquid. Because the dispersion points are not going to be broken up, because they have those long chain hydrocarbons and they don't have any bends in them. The only time they have a bend is when it bends with a double bond. And if they're saturated, they have no double bonds. So it's going to be a high melting point, high boiling point. They're going to be fat at room temperature, and they're going to stick in the bloodstream. Okay. So if your diet is high in saturated fats, you have high cholesterol level, you tend to have high blood pressure and other cardiovascular diseases. Although the relationship between saturated fatty acids and cholesterol is still not really clear, it's known that a diet rich in unsaturated fats results in decreased cholesterol levels. So unsaturated fats cause usually a decrease in cholesterol levels. Saturated fats cause an increase. Okay. The use of unsaturated fats in diet results in a decreased level of LDL, and an increased level of HDL. Okay. LDL is the bad cholesterol. HDL is the good cholesterol. Takes cholesterol out of the bloodstream. So with the positive correlation between heart disease and high cholesterol levels, The current dietary recommendations include a diet that is low in fat, low in animal fats, and the substitution of unsaturated fats like vegetable oils for saturated fats like animal fats. So that's how you should be getting your fat intake. It should be by unsaturated fats. What is the mechanism of uptake from the plasma? Endocytosis. The cell gets triggered by the LDL receptors, invaginates, okay, and then these LDL particles go in, and this thing actually folds around them and pinches off inside. Right? So the LDL binds to cholesterol molecule in the plasma. The LDL cholesterol complex then binds to LDL receptors found on the outer surface of the cell. The membrane then encapsulates, endocytosis, the complex, and it is brought inside the cell, endocytosis. Okay. The entire LDL cholesterol complex is brought into the cell, surrounded by a portion of the cell membrane, and this is called an endosome. And then the lysosome comes. Lysosome comes, fuses with that endocytotic vesicle, and then eats or destroys everything inside of it. That's how you get rid of it. What is the role of a lysosome in the metabolism of plasma lipoproteins? It fuses with the structure that's been for endocytosis, okay, and it starts to dissolve it. So lysosomes fuse with the endosome and release a hydrolytic enzyme. Okay. Why is my screen not updating? Okay. It cleaves that bond holding the cholesterol molecules to the LDL. Okay. Then it releases the cholesterol back into the cell. And the presence of cholesterol in the cell stops the cell from making more cholesterol because it already has some in there. Structure of biological membranes. Each type of cell has a unique membrane with varying percentage of lipids, proteins, and carbohydrates, making up your cell membrane. Currently accepted model of the membrane is a fluid mosaic model. The shape of the membrane is always changing. It's a lipid bilayer, which means the hydrophobic is on the inside, hydrophilic is on the outside, and the shape and composition is always changing. The amount of proteins in it, the amount of lipids in it is constantly changing. That's why it's called a fluid mosaic model. Okay, so biological membranes are lipid bilayers in which the hydrophobic and hydrocarbon tails are packed in the center of the bilayer, and the ionic heads are exposed on the surface to interact with water. So what you have is you have your hydrocarbon tails. Okay. So this inside section here is your lipid layer. Okay, that's your hydrophobic area. The outer portion is your polar head, that's amphoteric. So the hydrocarbon tails of membrane phospholipids provide a thin shell of non-polar material that prevent mixing of molecules on either side. It prevents molecules from moving. It's a membrane. The non-polar tails of the membrane phospholipids provide a barrier between the interior of the cell and its surroundings. The polar heads of lipid are exposed to water. They're very polar. They're soluble because they're hydrophilic, and they're very highly solvated. They interact with the molecules and dissolve within the polar solvent. The two layers of phospholipid body membrane are not identical in composition, okay, because of the proteins and the lipids that are embedded in them. Okay, in human red blood cells, 80% of the phospholipids in the outer layer of the membrane are phosphate acetylcholine and sphingomyelin. Okay, whereas phosphate diethylethaneamine and phosphoalylserine make up 80% of the inner layer. So the outer and inner layers are different because of the different cholesterol and the different proteins making up the different sections of that membrane. Carbohydrate groups are found attached to only those phospholipids found on the outer layer of the membrane. So the carbohydrates do not attach on the inside of the membrane, only to the phospholipids on the outer portion of the membrane, the side close to the external cerebral fluid. Here they participate in receptors and recognition functions. So they act to recognize certain types of things and how the cells perform certain activities. Okay. So this is your lipid bilayer. This is your hydrophobic tails. This is the hydrophilic group. And your lipid bilayer is formed. That's a much better picture. Is your bilayer. Hydrophilic is on the outside here. All these hydrophobics are on the inside. Okay. And then you get different groups embedding on them. You get the phospholipids, the cholesterol, the sphingolipids. Okay. Polar head, you have the polar groups on inside and outside the cell. But it's because that bilipid layer, remember, that protects everything, that keeps things from moving across. We can use some liposomes for delivery system. We're delivering different drugs. So liposomes were discovered back in 1961. During the studies of phospholipids and blood clotting, he found that if you get mixed phospholipids in water, tiny phospholipid bilayer sacs called liposomes would form spontaneously. They'll arrange themselves, though the hydrophobic parts are away from the water. They'll form micelles, just like we talked about. Okay? Since then, liposomes have been developed as an efficient delivery system for everything from anti-tumor, antiviral drugs to hair loss therapy, which is minoxidil. Okay? Okay? So we can deliver anti-tumor, antiviral drugs by using liposomes, by incorporating them inside that liposome. So they can travel through a polar compound like blood plasma, even though it's not polar, and then derive the site it needs to be at. So if a drug is included in the solution during formation of the liposome, the phospholipids will form a sac around the solution. And that's what stores it. In this way, drugs become encapsulated with a phospholipid sphere. So a non-polar drug can now be encapsulated inside one of these micelles, which is non-polar on the inside, but polar on the outside. So the outside can pass through the bloodstream and get delivered to where it has to go. These lysosomes can be injected intravenously or applied to body surfaces. Sometimes scientists include hydrophilic molecules in the surface of the liposome to help it get started. This increases the length of time that they remain in circulation in the bloodstream. Time-released medication works this way. They produce more of the lipids inside the membrane, so it doesn't dissolve as quick. And now you have time-release drugs. These so-called stealth liposomes are being used to carry anti-cancer drugs, such as doxorubicin and mitoxantrone. Liposomes are also being used as carriers for antiviral drugs like AZT. AZT is used in treatment of AIDS. It's the new drug for the AIDS treatment. And DDC, they're used to treat human immunodeficiency virus. Okay. So AZT is not soluble in blood plasma, but if we put a lysosome around it, now it's soluble in the hydrophobic part, and the hydrophilic part on the outside surrounding that molecule now can go through the bloodstream and be delivered where we need it, because there's receptors that release that into the cells that need it. So it's a way of delivering drugs. So a clever trick to help target the drug carrying liposome is to include an antibody on the surface of the liposome. Okay, these antibodies are proteins to bind specifically to the surface of a tumor cell. Antibodies are formed in infection, in response to an infection. Okay, if the tumor is an infection in the body, these antibodies bind to that cell to mark it as foreign. Okay, if we have those antibodies on our carrier, it's going to go to the tumor cell and bind to it, and we deliver our drug right to where we need it. And that's how we do it. Upon attaching to the surface of the tumor cell, the liposome membrane fuses with the cell membrane. This puts the AZT or the other molecule we're transporting directly into the cell that we wanted it to go into. In this way, the deadly chemicals are delivered only to those cells targeted for destruction by the antibody. This helps to avoid many of the unpleasant side effects of chemotherapy and occurs the moment healthy individuals or cells are killed by the drug. That's the big problem with cancer. A lot of the times we can't tell, because cancer is a disease where your own cells are multiplying at a very fast rate. So there's no difference between the cells of your body and the cells of the cancer or the tumor. So chemotherapy, a lot of times, if we can't make that distinction, we're just going to kill all those cells. And we're going to see what dies first, either the tumor cells of the cancer or the patient. That's what we do. That's why chemotherapy has such lousy effects on people. Hairs fall out, they get sick, they get miserable, they get nauseous all the time. It's because we're killing their body off. Okay, it's a race between what's going to die first, the cancer cells or the good cells. And there is no distinguishing factor because it is our own normal cells growing at a faster rate. So there is no antibody that's going to bind specifically to that one. Now, with AZT and stuff like that, with AIDS, there is a distinguishing factor because there's a virus inside there. Okay, now certain cancers we can do that with. Other cancers, we have no idea what the difference between the cells. There is no magic bullet that's going to only attack cancer cells and not touch human cells, healthy human cells. Because cancer cells are our regular cells, just growing at a faster rate. So another application of liposomes is in cosmetic industry. Okay, liposomes can be found that encapsulate a vitamin, herbal agent, or nutritional element. Okay. When applied to the skin, liposomes pass easily through the outer layer because it's hydrophobic. Okay, outer layer of dead skin delivers the content to the living skin cells beneath it. As with the pharmaceutical liposome, these liposomes, sometimes called cosmeceuticals, fuse with the skin cells. And they release their medication directly into the cell. Thus, delivery the beneficial cosmetic agent directly to the cells that can benefit the most for that drug, the underlying cells of the skin. Okay. So this is your water-soluble drug. Okay, here's your cell membrane surrounding it. This travels through the bloodstream, fuses with an existing cell by triggering the LDL receptors, and the material is put directly inside the cell where it belongs, where it can do the destruction or whatever what we need it to do. Okay. Most membranes require proteins to carry out their functions. We talked about proteins a lot already. Integral proteins are embedded in or extended through the membrane. Okay, so they go in the side or completely through one end to the other. Peripheral proteins are bound to membranes primarily through interactions with the integral proteins. So you have the integral proteins within the membrane binds to peripheral proteins on the outside. Membranes are not static. We said that several times. They're composed of molecules in motion. The membranes are always changing. That's why they call it the fluid mosaic model. It's like a fluid. The fluidity of the biological membrane is determined by the proportions of saturated and unsaturated fatty acid groups in the membrane phospholipids. Right? Remember, the saturated ones are going to stick together and make it less fluid. The more unsaturated fatty acids there are, the less they're going to stick together, the more fluid that membrane is going to become. Okay? About half of the fatty acids that are isolated from membrane lipids from all sources are unsaturated. Okay? Unsaturated means they have multiple bonds, means they're not going to line up and create those London dispersion forces, which means it's going to be fluid. It's not going to be like animal fat. The unsaturated fatty acid tails of the phospholipids contribute to membrane fluidity, like we said, because the bends introduced in the hydrocarbon chain by the double bonds. So if you have the bend, in other words, if you got a hydrocarbon chain and you got a hydrocarbon chain, they're going to line up. Okay. No double bonds, everything lines up. You get London dispersion forces, and it holds all these together pretty strong. Okay. Now, if you throw a double bond in there, okay, all of a sudden, we're not lining up here anymore. Okay. So now this is going to be more liquid. This is going to be more solid, like animal fat. Okay. So if it's unsaturated, it's going to be a liquid at room temperature, and a liquid in the body around the membrane. So if you have unsaturated proteins, okay, in the membrane, it's going to be more fluid. If you have saturated protein, saturated hydrocarbons, okay, it's going to be more solid. Okay, because the kinks of fatty acid tails do not pack together. In other words, these can't pack together like those can pack together. We also find the percentage of unsaturated fatty acid groups in membrane lipids is inversely proportional to the temperature of the environment. The higher the temperature, the lower the amount of unsaturated fatty acids we have. Okay, so bacteria have different ratios of saturated and unsaturated fatty acids in their membranes. depending on the temperature of their surroundings. Okay. The membranes of bacteria that grow in the Arctic Ocean have high levels of unsaturated fatty acids. So the membranes remain fluid even at freezing temperatures. Okay, so if they're in very, very cold environment, okay, they're going to have high levels of unsaturated fatty acids. Okay? That means even as things freeze and get really, really cold, their membranes are still going to move because these unsaturated fatty acids, double bonds that keep these the fatty acids from sticking, the hydrocarbon chains from sticking together. Right? So when it's really cold environment, you're going to have more unsaturated fatty acids in the membrane. Really hot environment, you have more saturated fatty acids because they're going to be liquid anyways. It's moving. Okay. So conversely, the organism that lived in the hot springs of Yellowstone National Park with temperatures near the boiling point of water have membranes with high levels of saturated fatty acids. Okay. Because they're going to stick together, but it's a very hot environment, so they're going to be boiling off and moving around. Okay. This flexibility in fatty acid content enables bacteria to maintain the same membrane fluidity over a temperature range of almost 100 degrees. So if one bacteria is at 0 degrees centigrade, one's at 100 degrees centigrade, they're still going to have the same flexibility. And that's what allows to happen because of the concentration of the ratio between unsaturated fatty acids and saturated fatty acids. Okay, bacteria growing in Yellowstone National Park are called thermophiles. Thermophiles because it's really hot in there. And temperature approaching the boiling point of water, what type of fatty acid do you think we found in the membrane phospholipids? All right. So what type of fatty acid we found? If you're in Yellowstone National Park and you're looking at that thing and that water is hot as anything, okay, what type of fatty acids would be most common in the membrane of those phospholipids of the bacteria?
Unsaturated?
It would be, no, really hot would be saturated, right? Really cold would be unsaturated because you want to move. Because it's going to be saturated fatty acids because saturated fatty acids have all single bonds and they're going to line up. So it's a really hot environment. So this is going to move anyways because of the heat. So this tends to make it the same consistency as one that's cold environment that we have unsaturated fatty acids. So generally the body temperature of mammals are quite constant and fatty acid composition of the membranes are usually very uniform with the membranes in the body. Okay. One exception is reindeer. That's because he's needed for Santa Claus's sleigh. Okay. Much of the year, reindeer must travel through ice and snow. Right? That's where they live. Thus, the hooves and legs must function at much colder temperatures than the rest of the body. So the hooves and the legs are under snow and much colder than everything else of the body. So the membranes, you would guess, would be different. Because of this, the percentage of unsaturation of the membranes varies along the length of the reindeer leg. So here's one species, okay, that if you take a sample at different parts of the leg, the saturation of the cell membrane is going to be different percentage of concentrations of saturated versus unsaturated hydrocarbons. Okay. So we find the proportion of unsaturated fatty acids increases closer to the hoof. So why is it not going across? Well, at least my connection, it goes. Okay. All right. So the proportion of unsaturated fatty acids increases closer to the hoof. As it gets colder and colder, then we have the more unsaturated fatty acid because those are the ones that are not going to stick together and need to move in a cold environment. Okay. This permits the membranes to function in low temperatures of ice and snow where the leg is exposed to. As you get away from the hoof and the legs, as you're more towards the top, you're going to have more saturated fatty acids because it's warmer up there. And they don't need all that double bonds to help prevent it from moving around. So it'd be the same function on everything. So membranes are fluid regardless of the environmental temperature conditions, whether it's really hot or really cold. We keep their fluidity by adjusting the concentration of saturated, I'm drawing blanks on words here, saturated versus unsaturated hydrocarbons. Okay. It's been estimated that membranes have the consistency of olive oil. That's what a membrane is like. That's how fluid it is. It's not the solid structure at all. Okay. Although the hydrophobic barrier created by the fluid lipid bilayer is an important feature of the membrane, the proteins embedded within the lipid are equally important and responsible for cellular functions, different critical cellular functions. So that lipid bilayer is important to keep things in and out, and the amount of proteins that are actually embedded in that membrane are also important. The presence of these membrane proteins revealed by electron microscope called freeze fracture. Cells are frozen, okay, very cold temperature, then fractured with a fine diamond knife. Once they're fractured, you can see between the two layers of the lipid bilayer of the membrane. You can pull it apart. When viewed with electron microscope, the membrane appeared to be a mosaic studded with proteins, like we thought. It's a fluid mosaic model, and all these proteins are stuck within it. Okay, because of the fluid of the membranes and the appearance of the proteins, our concept of membrane structure is called the fluid mosaic model. Okay, it means it organizes by itself into like micelles, and it's got the fluid like olive oil. That's how it moves, and there's proteins embedded in it. That's the mosaic portion. And this is what it looks like. This is a really good picture of it, because here's your polar heads, and all this stuff inside here. These are your non-polar or hydrophobic. And we have different proteins on this. So if you look at this close to, this is the non-polar regions, and this is the polar regions of our membrane. These are receptor proteins. They bind to areas which determine whether endocytosis is going to occur to absorb cholesterol or something else in there. These are peripheral proteins. Okay, this is a membrane channel protein where certain kind of ions and other type of small molecules can pass through this channel. Okay, we have carbohydrate chains. These are your sugar chains. Okay, we have glycoproteins that are embedded in the membrane, and glycolipids. This is the external membrane surface, the phospholipid bilayer, internal membrane surface. These are your peripheral proteins. Okay? You have cholesterol that's embedded in your membrane. This is a cytoskeleton inside the cell. That's what holds the cell as like a scaffolding to support it. And you have transmembrane proteins, which are proteins that bring stuff like glucose. You can bind to it, enclose in it, push it in, and bring it into the cell. So all these different things function within this fluid mosaic membrane for our membrane structure. So some of these other proteins called peripheral membrane proteins are bound only to one of the surfaces of the membrane by interaction between ionic head groups of the membrane lipids and ionic amino acids on the surface of the peripheral protein. So depending on what they're binding, those proteins are acting a different way. Other membranes, proteins called transmembrane proteins, are embedded within the membrane, okay, and extend completely through it. So some are only on the outside, some are on the inside, and some go completely through it. Okay, so different types of proteins have different functions, and it acts like that depending where they're located on the cell membrane. So it's the ones that go completely through it are exposed to both inside and outside the cell, because they're going completely through the lipid bilayer. Just as the phospholipid composition of the membrane is asymmetric, so too is the orientation of the transmembrane proteins. They don't line up exactly. They're just randomly placed throughout the cell. So each transmembrane protein has a hydrophobic region because it has to go through the hydrophobic area of the membrane that's associated with the fatty acid tails of the membrane phospholipids. So it embeds in the middle of the lipid bilayer. Each also has a unique hydrophilic part that's found associated with the outer layer of the membrane as located outside the cell. So it's amphoteric also, it has to be, so it can bind to both areas. The region of the protein typically has what's called oligosaccharides covalently attached. Those are sugar units, 3 to 9 sugar units are oligosaccharides. Hence, these proteins are called glycoproteins for your sugars and your proteins attached to them. Similarly, each transmembrane protein has a second hydrophilic domain that's found associated with the inner part of the membrane and projects into the cytoplasm. So remember, if your cell membrane We'll just do a little cell membrane here. Fatty acid tails. Okay, there's your cell membrane. Hydrophobic on this side. If a protein extends both sides, it has to have a polar region here, a polar region here, and a hydrophobic or non-polar region in between. It has to have two polar ends, one on the outside of the cell and one on the inside of the cell. And then inside the membrane, it has to be non-polar or hydrophobic. So it can exist both places. Those are trans proteins that go from one side to the other through the cell membrane. Typically, this region of transmembrane protein is attached to the filaments of the cytoskeleton, and that's what holds it in place once it's inside the cell here. Okay, so membranes are dynamic structures; they're not static. Membranes are constantly changing. Okay, and we'll finish up in just a little bit here. So the mobility of proteins embedded in the biological membranes are studied by like labeling certain proteins in human and mouse cell membranes with red and green fluorescent dyes. So you can label them and you can see where they go as they change. The human and mouse cells are fused together, okay, cause the membranes of the mouse and human to flow together to create a single cell. Okay. The new cell was observed through a special ultraviolet fluorescent microscope. The red and green patches were localized within the region of the original cell membranes when the experiment began. And 40 minutes later, the color patches were uniformly distributed in the fused cellular membrane. So you took a human cell and a mouse cell, and you label the proteins with radioactive or fluorescent dye. Then we fused those together. Then we took a look at it, and all the proteins were right where we labeled them. Give it 40 minutes, and they all dispatched evenly through it. So everything spread out evenly, which goes to show that it's dynamic and constantly moving to a random position. Spread out. So the experience suggests that we can think of the fluid mosaic membrane as an ocean filled with mobile floating icebergs as it gets changed positions through there. Okay, so here's your mouse cell, here's your human cell. They fuse together, okay, and everything was right where we put them in. But after the fusion, about 40 minutes later, all randomly associated. Everything randomly distributed across it. So the membranes are constantly moving to the fluid mosaic model.